Hello, my name is David Ehrman. I am here in Springtown, Texas, and what I wish to discuss today is scientists who confront evolution, who confront evolutionary proponents. This book was written by Henry Morris and Gary Parker. I know that Henry Morris is deceased. I'm not sure about Gary Parker, but they were creation scientists, and often I don't reference young Earth creationists, but in this particular case, I will. On this particular topic, I will. And they are discussing some evolutionary proponents who wrote this book called The Structure and Action of Proteins. These evolutionary proponents were Richard Dickerson and Irvin Geis, spelled Geis. And this is from Morris's and Parker's book, What is Creation Science? Copyrighted 1982-1987 by Muster Books Incorporated, 19th printing, July 2004. And now I am going to quote from the book by Morris and Parker. From the perfection of protein sequence and structure analysis, we can pin down with great precision the relationships between the species and how the proteins evolved. Then with every example, they, who are Dickerson and Geis, they proceed to disprove that evolutionary prediction. Consider hemoglobin, for example, the protein that carries oxygen in red blood cells. Dickerson says that hemoglobin poses a puzzling problem. Hemoglobins occur periodically among the invertebrate phyla in no obvious pattern. That is, I would suggest they occur in a creationist mosaic or modular pattern, like bits of blue-colored stone in an artist's mosaic. We find hemoglobin in all vertebrates, but we also find it in in nearly all vertebrates, but we also find it in some annelids, bracket, the earthworm group, close bracket. Some echinoderms, bracket, the starfish group, close bracket. Some mollusk, open bracket, the clan group, close bracket. Some arthropods, open bracket, the insect group, close bracket, and even in some bacteria. In all these cases, we find the same kind of molecule, complete and fully functional. As Dickerson observes, it is hard to see a common line of descent. Smoking in so snaking in so unsystematic away through so many different phyla. If evolution was true, we ought to be able to trace how hemoglobin evolved. It does not seem possible, he says, that the entire 8-helix folded pattern appeared repeatedly by time and chance. As far as creationists are concerned, hemoglobin occurs complete and fully functional wherever it is appropriate in creation, somewhat like a blue-colored tile in an artist's mosaic. And that's from pages 57 and 58 of Morris's and Parker's book, What is Creation Science? And that will be enough for today. Thank you for listening, and please check back again.